lesions in pre-surgical patients with epilepsy. So really the aim was to improve the detection of otherwise subtle lesions that might be overlooked on clinical scans. And yeah, this is just a teaser. I'm going to talk about this patient later, but it looks like it might be working. And first of all, I will have to, I'd like to introduce you to epilepsy surgery and as to why neuroimaging might be important there and which diagnostic gaps need to be filled here. So around about 1/3 of patients with epilepsy will not become seizure-free with anti-seizure medications. And in a lot of these patients, epilepsy surgery can be like a curative option, which is pretty amazing actually. But quite logically, I think the rates for success are a bit higher if there's a lesion present on preoperative imaging. So if you know where to cut, the patient will have a higher likelihood of becoming seizure-free. And the problem is that around about 1/3 again of these patients will not have a lesion on clinical imaging, neither on FDG PET, where we'd expect a hypometabolism, or on MRI. So this, at least I think, is one contributing factor to very stable rates of seizure freedom postoperatively over the last 3 decades. So despite technological advances, there has not been a lot of improvement there. So this is why we believe new modalities to guide surgery, but also electrode implantation would be crucial. And I'd like to briefly touch upon stereo EEG invasive implantations, because this is going to be the reference standard for our study to relate our microstructural changes to. So as you can see here, there are these little shafts or rods implanted into the patient's brain with lots of electrode contacts along the way. And these electrodes are going to be implanted. The patient is going to come to the ward, and we are going to take away their anti-seizure medications and try to record as many seizures as possible in the aftermath, so we can just pinpoint the locations where these seizures are coming from invasively, and also to perhaps tailor a resection margin for later surgery. We can also apply like a current to these electrodes to map function of the tissue, which is actually pretty cool. And next, I'd like to talk about this little image over here, which I have stolen, because while many of these lesions are subtle on clinical imaging, it is generally, most of these lesions are very well defined on histology. So this example is a focal cortical dysplasia, where you can see in orange, these orange blobs are, where is it, the cursor? Here, are really huge balloon cells, which are dysplastic, and in green you can see heterotopic, that is cell bodies where they should not be, heterotopic cell bodies in the white matter, which are also enlarged and dysplastic. So diffusion MRI might be a suitable modality to probe and to actually visualize these changes in these lesions, for example, with the Sandy model that has been touched upon previously, where we can model or try to image cell body signal fractions and also apparent radii of these cells. So I'll skip this. So our hypothesis was that lesional tissue that generates seizures might show these microstructural alterations, and that we could leverage these alterations to detect these lesions and also characterize them. So these are our methods. I go over them very quickly. We conducted a prospective study with this invasive reference standard. So prior to implantation, we got these patients into this Connectome 2 scanner, and we managed to include 13 patients and 33 healthy controls, so that we had a very well characterized cohort with this invasive reference standard to relate our microstructure to. Just very briefly, or you can see the protocol over here. And so we fitted the Sandy model in each patient and in each control, and we obtained these maps of soma, neuroid, and extracellular signal fractions, and these apparent soma radius. And since we aimed at individualized lesion detection, we chose a normative mapping approach, where we can, at each vertex position, compute a Z-score compared to controls. But more importantly, on the right over here, you can also compute a multivariate score of deviation relative to controls, which is this Mahalanobis distance measure, where you just combine all 4 parameters to compute this individualized deviation map, which can then very neatly be used to relate these changes to invasive electrophysiology, as shown in this patient over here, or to probe the biological or metabolic relevance of our microstructural changes relative to FDG PET uptake. Okay, so I'd like to start first by showing some group results to show that at the group level there's an added value of microstructural imaging in detecting these reference standard epileptogenic regions. So very briefly, you see here the post-SEEG reference in this one example patients where both temporal lobes were involved. We have a clinical prediction at the lobar level right now, very coarsely. In this case, none. And then we just blindly selected the lobe with the greatest microstructural abnormality on average and just computed the incremental hit rate or sensitivity to this post-SEEG reference, and SEEG being the implanted electrodes. So as you can see here, we have, at this point, not all patients have been implanted yet, but we did observe a significant increase in recall to these, or in sensitivity to these abnormal regions of 0.2. And this was driven by mainly 3 subjects. But more importantly, which is pretty amazing, I believe, is that even when there was no increment over clinical imaging in sensitivity to these regions, all these dots are green. That means the blind prediction of this most abnormal lobe was correct nonetheless. So that is pretty nice. And one more group analysis before I'm going to show some individual patients' results. Then we investigated whether we can classify at the ROI level regions based on this microstructural deviation, whether they are seizure onset regions or not, red being seizure onset regions. And this is also possible with moderate area under the curve for 2 different parcellations. So we can discriminate between seizure onset regions and non-seizure onset regions just by blindly looking at the average microstructural deviations from controls. And this, I think, is a bit of a conservative estimate because of the sampling of our SEEG electrodes. You have seen we cannot sample the entire brain with it, so we have very sparse sampling. So I want to show, or I want to highlight this patient, subject number 2, where the actual lesion has not been implanted. So this is a patient with right temporal lobe seizures. And you can see here on the right side a hypometabolism on FDG PET, where you nicely see the difference across both hemispheres. So here we detected a very small and circumscribed cluster of abnormalities in the ventral temporal pole. And these blue dots are the horizontally implanted electrodes, which do not cover this region. Here you can see the implantation scheme down there, where the seizure onset or the first seizure activity was actually recorded in the right amygdala. So before I go over, oh no, I don't know how to go back, so I don't go ahead now. So the patient has been resected. Luckily, not only the amygdala has been resected, but also the temporal pole. And on histology, the amygdala was completely normal, but the ventral temporal pole nicely showed a neuronal white matter heterotopia, that is cell bodies where they should not be in the superficial white matter, and also cortical gliosis. And now I'm going to show you Z-maps for this little cluster. I hope you can see it even. So first of all, this is the same map, this Mahalanobis distance map on the surface, thresholded, that is FDR corrected, that is very specific to this little small cluster over here. And when looking, for instance, on the superficial white matter, you can see a decrease in neurite signal fraction over here and an increase in soma signal fraction over here, which contribute to this abnormality that we saw multivariately over here. So this nicely matches with our histology, where there are cell bodies where they did not belong, decreasing the neurite signal fraction. I don't know how much time I have left, but 2 minutes. Okay, I'm gonna hurry. This is the patient from I showed in the beginning. Also right temporal lobe epilepsy. At the time of the study, MRI and FDG PET negative. And this is the T2 flare scan, which is a commonly used, oh, how do I go back? commonly used structural sequence in the evaluation of these patients. And here you see the raw soma radius map threshold between 8 and 10 micrometers, where you see this nice little increase in soma radius, which, when we look at the z-scores, is actually increased relative to controls. And combining these metrics multivariately show a nice deviation in the right hippocampus. And relating this to invasive electrophysiology, we saw that this increase or this deviation was located in the seizure onset zone, which has been implanted, and that these changes, these microstructure changes, nicely aligned with FDG PET hypometabolism, which we can even see when we visually over here. Okay. And this patient had hippocampus sclerosis on the right side on histology. That is a common epileptogenic lesion. So I'm going to skip a lot of subjects now to show you the first patient we have scanned on the CMA-X to show some clinical translation. This is a patient with invasively 3 seizure onset zones, one in a parietal cystic lesion, which has been thought to be perhaps a post-ischemic lesion first, and both hippocampi. All have been implanted, and we don't have a control group yet for these patients, but for this scanner, but just looking at the soma radius map, we can already see an increase relatively to the surrounding gray matter in soma radius in all 3 seizure onset regions, and especially so in this cystic lesion, which turned out to be a ganglioglioma, of which we know that it contains large ganglion cells, which are very huge. So we nicely captured this microstructural characteristic. And yeah, I think then I'd like to conclude, and I can show more patient data later if anybody is interested. So yeah, in this prospective study, it turned out we can detect these epileptogenic lesions irrespective of their of the etiology, because this was a very heterogeneous cohort, and the Sandy microstructure modeling did indeed add incremental localizing value for these seizure onset regions. So I believe that this should be a modality that should be investigated further in these patients in perhaps larger studies, and also in more clinically feasible models. For example, the clinical Sandy model might be primed for this. And yeah, just on the right side, you see a nice patient with tuberous sclerosis. Each of these lesions is a cortical dysplasia-like lesion of which we know histologically that they have large soma radii. And this is nicely reflected in this R-soma map right here as compared to the FLAIR image. So yeah, thanks so much for the attention. And yeah, I'm looking forward to your questions.